So this session is the MyHeritage DNA 101. I'll go over the basics. I'll explain some of you probably, who has taken a DNA test? Whew. All right. So we'll go over the basics. Some of you have taken a DNA test. Some of you already reviewed the results. We'll explain a little bit behind what's going on behind the scenes, how it works, how we're doing that matching magic that we do, how the ethnicity estimate is calculated, and it'll be fun. Join me. All right. So the basics, even before we are able to regenerate Herzl's original DNA, there are 22 autosomal chromosomes, pairs of chromosomes, and an additional sex chromosome which determines our sex, and X and Y for males, and double X's for females. And basically all the information is hidden right in these, in each one of us. And this is the kind of thing when you take the DNA test, you send it to the lab, and I'll show you how it eventually looks like. This is what we're looking into. This is where all that secrets, all these matches and ethnicities hidden inside. So there are over 3 billion base pairs in those 23 chromosomes. But we don't need to look at all of them. That would have taken us a lifetime to calculate matches for every one of the SNPs, those genetic markers that exist in our chromosomes. And I like this example. I use it quite often. So we'll do it again. In this picture, you see two genomes on the top and on the bottom. They look quite the same. But when we look deep inside, the one at the top is of a human, while the one at the bottom is of a chimpanzee. So you can see that there's a lot of resemblance. A lot of things look quite alike. And actually, we're 98% similar. Our DNA looks exactly 98% exactly the same as a chimpanzee. When it comes to human beings, the number goes up. We're 99.9% similar, those 3 billion pairs, almost identical. What we're left with is that 0.1% that distinguish us one from the other and makes us those unique us that at least I'd like to think that I am. So the ethnicity estimate. The definition that we go with is the architecture of genome variation between populations. Basically, our attempt to distinguish and say, you're from this ethnicity and you're from that, or from a combination of a couple of these, right? And let's see how it works. We won't use specific ethnicities for this example. We'll go with colors. So this is Mike. And Mike has a maternal side that looks something like this. And let's say that his great-grandparents from the maternal side, all of them are purple. They're coming from the purple population. My favorite one. So if all of his great-grandparents are purple, so will be his grandparent and his grandmother, his mom, and therefore eventually Mike will be 50% purple. All right? So this is the easy side of the family. Because on the paternal side, things are a bit different. So here we have two great-grandparents who are orange, and one who's red and one who's blue, right? So as we go down in the generation, we inherit about 50% of the ethnicity from each one, depends on which kind of segments we've inherited. So that's like his grandparents, his maternal grandparent is grandma is orange because both our parents are orange. But the other side, you can see that his grandfather is already half blue and half red because he inherited approximately half from each side of his parents, right? And when it comes to his father, it's already 50% orange and then 25% of each. And eventually when we get to our dear Mike, he ends up with 50% purple, 25% of orange, and then 12.5% of the blue and the red. And this is, in a nutshell, how we're doing the ethnicity estimate, is that we're trying to determine those different ethnicities, different populations, not really giving them colors, but giving them names, and then looking at your DNA results and trying to say, what do you have in your inheritance? So we've conducted a project at MyHeritage. We call it the MyHeritage Founder Population Project. And we started by looking at trees. We have millions and millions of trees at MyHeritage, and we were looking for those people who have long chains of ancestors coming from the same geographical location, for example, or bearing the same surname for a long chain of ancestors. And we spotted them, we marked them, and we said, these are good potential to be a representation of an ethnicity, let's call it for now Scandinavian. I think you're familiar with that. So we started by that, and we started sending samples, more than 5,000 of them, to different participants of the Founder Populations Project. And we started sampling them and trying to come up. And then when we were looking at their DNA, we were trying to look for a signal, something that kind of repeats itself, right? If we have 10 people tested, we're looking at those 10 people, and we're trying to see whether we have something that stands out, something that's repetitive for every one of them, so that we can say, this looks like a Scandinavian guy, or this looks like an Iberian person, all right? Those pictures were taken by us at MyHeritage. We have a wonderful project I recommend you to look at. It's called Tribal Quest. We're going all around the world. We're helping tribes from deserted places document their family history. We're building family trees for them. We're sitting with them. We're meeting with them. Kind of a side note, I recommend you all to look at it. But we're also taking DNA tests and trying to preserve that unique heritage. These are not people that travel too much around the world. They usually stay at the same place for a very, very, very long time, so that we can take that DNA test and we can tell people from various places around the world, you've got some, right there on the right, this is a Nenets tribe in Siberia, for example. So you've got a little bit of Nenets in you, or Namibia, or the other places that we went to and will continue to go to. All right, so we've collected all that information, all that the Founder Population Project came to an end. There's no real end here, right? Keeps on growing, we keep on learning, we are able to improve our algorithms, collect more data, and build more focused groups. So we're done, and now you're taking a DNA test. And this is actually what's being done. So after we've collected the founder population data, we build those models, those signals. We say, okay, in chromosome 7, we see that pattern that usually resembles a person from Scandinavia. So when you take your DNA test, we compare your results against those models. And we're trying to look and try to break down your own ethnicity into, give you an ethnicity estimate result. And we aggregate it and compare it, and eventually we're giving you your ethnicity estimate report, which consists of those ethnicities that we have. At the moment today, we have 42 regions at MyHeritage. They're listed right here. And some surprises are expected soon. All right. So now let's talk about DNA matching, okay? And my example of Frank and Molly. So we have 22 autosomal chromosomes. I'm getting away from the mic. We're having 22 autosomal chromosomes, and in this example, I'm kind of want to show you what happens as generations go by, and what happens to the DNA. And in this case, I'm taking for reference think of those two lines that you see at the top for Frank and Molly as chromosome number 5. So this is how Frank's chromosome number 5 looks like, and this is Molly's. His is purple, hers is orange. Again, we're using colors, easier to understand. And remember, there are pairs. Each one of us, for each chromosome, we get one of the pairs from our father and one of the pairs from our mother. And that's exactly what we see in Ben's and Lisa's case. Now you can see that Frank has a dark purple and a light purple, and there at the middle, at Ben's number 5 chromosome, you can see that the color changes. These are the recombination points. So when the two pairs are recombining together and then switching between one and the other, that can happen more than once when Ben was conceived. Same thing like Lisa had 2 recombination points. So her one pair of chromosome 5 is a mixture of both her father Frank, his father Frank, and the same thing goes with the mother. So this is eventually how Ben's and Lisa's chromosome 5 looks like, okay? And when we're trying to compare, we're looking at those places on the chromosome which are identical. We look at them, we look for long segments, okay, that kind of repeat themselves. So we can see on the right that Ben and Lisa share this amount of DNA on chromosome 5. This is the amount of shared DNA that they have. And then later on, Ben and Lisa are getting married and having their own kids. and new colors come into play. So for Eric, we have the new blue color, okay, which came from Ben's spouses. And same thing for Lisa with the daughter Julia. And when we try to compare Eric and Julia together, now we see that things are a little bit different. And we see that there's even less shared DNA between them. And that's why, and Gilad spoke about it in his presentation, as generations go by, we lose the amount of shared DNA between people with common ancestors. Okay? Because Eric and Julia's common ancestors are Frank and Molly. So they'll be sharing roughly about 25%. But Ben and Lisa had approximately 50%. And that's why it's so important to test the more elderly relatives. And we'll get to that also in a second. So let's look at the report, because eventually when you're going online and you're looking at your DNA matches, you don't see Ben and Lisa. We haven't added them to the product yet. But you see your DNA matches, the people with who you share some amount of DNA. And we're telling you how many segments and what is the length of the, how many centimorgans, the length of shared segments that you have with them. And a percentage, that's what most users look at, how much shared DNA you have with one person or the other. So when it comes to 50%, that probably means a parent or a child or a sibling. 25% is taking you one hop away, and now you're talking about grandparents or grandchilds. At MyHeritage, we use different algorithms to rule out some options. So for example, if you get a 25% match with someone, it can mean that and let's say that he's a male, also something that we're taking into consideration. Let's assume you have a 25% match with a male. He can either be your grandparent or your grandson. But we're also looking at the age. So if someone signed up to MyHeritage and logged his year of birth, and we can see that he's older than you by approximately 50 years, it probably means that he's your grandparent. There's no point in even mentioning the grandchild. Right. So 25 and 12.5 and so on and so forth. So as the amount of shared DNA, the percentage of shared DNA decreases, it probably means we're getting a little bit farther away. Our common ancestors, our most recent common ancestors, are a little bit farther away from us. All right. And that's exactly what happens in our case. This is my tiny bit of my family tree on MyHeritage. Right next to me is my beautiful wife, Julia, and the youngest genealogist to live on Earth as of date, my daughter Mai. She's 3 years old. I tested her for DNA at the age of 2 approximately. Cost me a box of candies, but it went fine. And it kind of comes back to a question that was asked before, because people are asking me, but why are we testing your daughter? Because you have your DNA. I've tested my DNA way before I tested her. But her mother didn't want to test hers at some point. So if I'm testing my daughter, I can get my wife's DNA matches. Don't worry, she's tested since then. She's very happy. She's excited with the results. But when Mai actually gets a DNA match with a Jane Doe, and there is a segment, an IBD segment, it's an inherited by descent. There's a common ancestor that sits right there, up there at the top. We need to work hard to find that common ancestor in many of the cases. And I'll talk about it also in this presentation, but mostly in the next one. It probably means that up there, there's a common ancestor. And right here, it's over there at the blue one and the purple. They're both her ancestors, the common ancestors. And the DNA diluted. With the generations, it kept on passing by for the grandfathers to the father, and eventually to my daughter. And so the same thing happened with Jane. Okay? All right. So now let's take it a step further and talk a little bit about, okay, you've taken a DNA test, and it goes to the lab, and that's the toughest time. You wait, I know, for 2, 3, 4 weeks sometimes. And finally, the results are ready. And it's great. They're done. The lab is done, and we're ready to present you with your results. And it looks like that. That's what comes out when you take a DNA test. It's not, no more nice pictures of those people from the Founder Population Project, right? So this is your DNA. This is basically what comes back, those 700,000 SNPs or so that we're testing. And then we're looking into those. So what you can see there are the chromosome. It's a very long list, 700,000 lines or so. And for each genetic marker, we look at what the result, what did we find in your DNA test. There are 4 letters, A, G, C, and T. The I represents a no call. We weren't able to find it. So this is what we have to work with. And from that, we need to find you some DNA matches and give you some ethnicity estimate. That's not easy. So we're doing a couple of things. And I'll focus about the first two, phasing and imputation. And I hope that this might be able to clarify some of the points that I spoke about previously. So what is phasing? Phasing is our attempt, as you saw in the file, there are two letters. And the reason there are two letters is one you got from your mom and one you got from your dad. To do DNA matching, we need to separate. We need to try and understand which one came from the mother and which one came from the father. And that will help us find DNA matches for you because we're looking at a single line, right? So phasing is our attempt to break that. When we see that you have, if I'm the child in this case, a BB and AA is trying to figure out and split those, and we're doing that by smart algorithms that can help us break those down and understand better which letter came from the mother and which letter came from the father. There are some cases in which we can't tell that, and we're using some statistical algorithms to figure that out. All right, so we narrow it down. We're done with the phasing. We have the first pair and the second pair. At MyHeritage, we allow uploads from other vendors as well. So if you've taken a test with someone else, we encourage you to upload your results. Therefore, this is free up till December 1st, where things are a little bit going to change. So I welcome you all to come and upload your results if you've already taken a DNA test. And the thing is that different companies look into different SNPs, different genetic markers. And that kind of makes our life hard because it's not apples to apples. We can't compare one DNA result to the other and find good DNA matches because there are some differences. So we're doing something that's called imputation. So here's an example where you can see that the first SNP exists in both samples, right? But the second one is missing and vice versa. So we need to fill those lines. We need to understand what was the result there so that we can give you good matches. And the best way to think about how we're doing that is by doing fill in the blanks. Now, how many of you can read the sentence that appears on the top of the screen? All right, good. But some letters are missing. You don't see them, right? And that's exactly how we're doing imputation. So your brain can perform amazing statistical calculations and fill in the blanks, even though some is missing. And that's what we're doing for DNA. We look at what's surrounding, like the results, those SNPs that we were able to see results for, and we look and we use statistical algorithms to fill in the blanks, to just, if there was a sequence of A, G, C, T, C, T, C, then usually the next one will come at T. And that's how we're filling the blanks, and we can do matching between different vendors, and that's something that's unique to us. And we're looking for those sequences that repeat, okay? All right, starting to run out of time. And these are the IBD segments. So once we're done phasing and once we're done with the imputation, we're looking again at these results. And we're taking the database. We have nearly 9 million people tested on MyHeritage. And we match. Every time a new kit comes in, we're matching it against the database and see if we can find those segments, long enough segments, that we can say that there's a potential good match here. There is a potential common ancestor for these two individuals. And we're looking for these segments, and once we can find them, this is what we add to the application. And this is what we show you as your DNA matches. I think that that's it for now.